So for the next, I'm going to introduce you to our very own Director of the School of Biological Sciences. Do the welcoming, Professor Lesego Molalekton. Professor, over to you.
Thank you, Chair. Good morning, colleagues, students, and guests. On behalf of the Microbiology Subject Group, the School of Biological Sciences, and the Unit for Environmental Sciences and Management, I warmly welcome you to our 2025 World Antimicrobial Awareness Week event. Now, as a subject group, WAAW is one of our flagship community engagement projects as a facet of our research focuses on AMR. And through this research, we continue to seek the understanding of AMR in our environments. And also through this research, we have been able to propose recommendations as well as strategies aimed at slowing the rapid spread of AMR within our environments. And not only that, but the outcomes of our research has also contributed greatly to strategies aimed at strengthening South Africa's as well as Africa's response to this growing global health threat. Now, today presents itself as an opportunity not only for us to learn, but to explore data-driven approaches that can help combat AMR. With that said, it is my absolute pleasure to also welcome our speaker, Dr. Carroll from Umeå University in Sweden. Dr. Carroll, thank you for joining us. We are looking forward to the lecture. Thank you, Chair.
I will now call to Professor Gaugelo Lekota to introduce the speaker for us. Thank you.
Good morning. Good morning, everyone, ladies and gentlemen, distinguished guests, colleagues. As we gather during World Antimicrobial Awareness Week, a time dedicated to strengthen global action against antimicrobial resistance, it is my honor to introduce a scientist whose work is shaping the future of pathogen surveillance and responsible antibiotic use. Please join me in welcoming Dr. Laura Carroll, an assistant professor in the Department of Clinical Microbiology at Umeå University and a principal investigator at the Umeå Center for Microbial Research. Dr. Carroll is a fellow of the prestigious Science SciLife Lab and Wellenbeck Data-Driven Life Science, which is also noted as the DDLS Research Group, so where she leads cutting-edge research in microbial research. Her academic journey from Michigan State University through a PhD at Cornell University to postdoctoral training at EMBL Heldenberg reflects her deep expertise at the intersection of bioinformatics, pathogen genomics, and public health microbiology. Dr. Carroll's research focuses on developing innovative methods for understanding pathogen evolution, outbreak detection, and antimicrobial resistance using phylogenomics, machine learning, and multi-omics approaches. Her work contributes directly to global efforts to detect, track, and combat resistant organisms. So that makes her presence today very special. So in recognition of her groundbreaking contribution, she received the Future Research Leader Award from the Swedish Foundation for Strategic Research. So today she brings her expertise to help us reflect on how data-driven science can advance global AMR surveillance and strengthen our fight against antibiotic resistance. Ladies and gentlemen, please join me in welcoming Dr. Laura Carroll.
Wonderful. Hello, everyone. It is an absolute pleasure to be here today. Let me just get my PowerPoint set up. This is the hardest part of any talk. Hopefully you guys can see the correct view and not my presenter view. That would be very embarrassing for me. But it's such a pleasure to be here today. Thank you to Professor Lakota for the absolutely wonderful introduction. Hopefully I can live up to that. I don't know if I can. So I really appreciate the kind words. As you guys already know, I'm an assistant professor at Umeå University in northern Sweden. My name is Laura, and it is such an honor to be here with you today, virtually at least, sadly not in person. But we're going to discuss arguably one of the most important topics in the world right now, which is antimicrobial resistance. But before we jump into antimicrobial resistance, I just figured I'd quickly introduce myself. I'm a bioinformatician, and my research group is interested in how the interplay between humans, animals, and our environment contributes to the spread of bacterial pathogens. So basically, we take massive amounts of genomic and metagenomic sequencing data, so basically trillions of A's, C's, G's, and T's, and then we use that data to monitor how bacterial pathogens spread, how they evolve, and how they cause illness in humans and animals. And if there isn't a method out there to do what we want, because we are software developers, we actually try to develop it ourselves. So I'm super lucky. I get to work on a ton of different really cool microbiological problems. But today I'm really excited to talk about one of my favorite topics, which is antimicrobial resistance. So antimicrobial resistance, or AMR, is one of the biggest threats to global public health today. If you don't believe me, you can take the World Health Organization's word for it. And each year, antimicrobial resistant, or AMR, microorganisms are responsible for millions of illnesses, thousands of deaths, and massive economic losses. By 2050, it's been estimated that AMR could be responsible for nearly 2 million deaths per year, and it will be particularly devastating for those age 70 and older. Further, in just 3 decades, AMR infections could kill as many as 10 million people, which is on par with the 2020 death toll for cancer. And sadly, this burden is not going to be imposed on all of us equally, as the majority of AMR-associated deaths are predicted to occur in Asia and Africa. So AMR is a huge deal. We're dealing with it right now, and it's only going to become more pressing in the future. So what can we actually do about it? Well, the World Health Organization asserts that there are 2 things that we can do as scientists. First, we can improve the surveillance of antibiotic-resistant infections. And second, we can invest in research and development of new antibiotics. So today I figured I'd just talk about how my research group specifically tries to tackle these 2 major problems. Emphasis on tries. Sometimes it's quite hard. But I'll start with some case studies showing how we can use genomic and metagenomic data for pathogen surveillance, including some work that we've actually been doing with some really awesome scientists in South Africa. So specifically, I'm going to be discussing the surveillance of antibiotic-resistant non-typhoidal Salmonella enterica. So non-typhoidal Salmonella is a gram-negative zoonotic foodborne pathogen that causes salmonellosis. And symptoms of salmonellosis include diarrhea, fever, abdominal cramps, and vomiting. And notably, Salmonella is estimated to be responsible for more than 93 million illnesses and 150,000 deaths globally each year, meaning that it's a serious global public health concern. And when Salmonella becomes resistant to antimicrobials, things can become even more complicated and even more dangerous. Specifically, antibiotics such as ciprofloxacin, azithromycin, and ceftriaxone are needed to treat severe Salmonella infections. However, when an infection is caused by AMR Salmonella, it can be much more difficult to treat, and this can lead to more severe infections and higher hospitalization rates. And so because of this, the US Centers for Disease Control and Prevention, or US CDC, they actually consider AMR non-typhoidal Salmonella to be a serious threat to global public health. So because it's such a serious threat to global public health, it's really important for us to understand which Salmonella strains are resistant to antibiotics, which antibiotics these strains are resistant to, and where these AMR strains are circulating in the world so that we can develop better strategies to combat their spread, as well as treat the infections that they cause. And a high throughput data-driven way to do this is to use whole genome sequencing, or WGS, data derived from Salmonella. So WGS is really cool because it gives us high resolution insights into the evolution, epidemiology, and AMR potential of Salmonella. And public health agencies all around the world are sequencing hundreds of thousands of Salmonella genomes from tons of different sources, including human clinical cases, animals, foods, environmental sources, you name it. And a lot of this data is actually made public, so anyone can use it. So we can use all this publicly available whole genome sequencing data from Salmonella strains collected from all over the world from a ton of different sources to actually track the spread of Salmonella and the antimicrobial resistance genes that it carries. So to illustrate how we can use whole genome sequencing data for Salmonella and AMR surveillance, I figured I'd mention two case studies that my lab has recently worked on, and these both actually took place in South Africa. So this first case, I was super lucky to collaborate with Dr. Itumeleng Matel, who's a really awesome food and veterinary microbiologist. Itumeleng and his team do a ton of amazing surveillance work across the whole country of South Africa, not only in things like livestock, but also in companion animals, wildlife, and foods as well. So basically, he and his team designed the study that I'm going to talk about right now. They were the ones that designed it, they led it, so they're the real stars. I just played a really small role in the data analysis, so all credit goes to Itumeleng and his really awesome team. But Itumeleng and his team had sequenced 63 Salmonella strains that they had isolated from animals and animal products in South Africa. And their goal was to kind of figure out which strains are circulating among animals and foods in the country and could pose a threat to South African consumers, and also if these strains are resistant to antimicrobials or not. But in addition to the 63 South African Salmonella genomes that Itumeleng generated, there's also thousands of publicly available Salmonella genomes out there that we can use. So we compared our 63 Salmonella genomes from South Africa to thousands of closely related publicly available Salmonella genomes collected from all over the world from a ton of different sources. And in this specific study, we tracked a couple different serotypes or major lineages of Salmonella. But in my opinion, the most interesting one turned out to be a serotype called Salmonella Dublin. So this is a very overwhelming phylogeny of Salmonella Dublin, basically all the Salmonella Dublin genomes ever sequenced at the time of this study. But don't be scared. So here we just have our phylogeny. And then here we have the time in years plotted along the x-axis. And then the tip labels of our phylogeny are colored by the continent from which each strain was isolated. And then over here, we have this really overwhelming heat map that shows the presence and absence of plasmids in blue and AMR genes in orange. So don't worry if you can't read the labels or anything. Just note that if there's dark blue shading, that means a particular plasmid was present. Light blue shading means that particular plasmid was absent. And the same goes for AMR genes. If there's dark orange shading, that means that particular AMR gene was present. Light orange shading means that particular AMR gene was absent. So there's a lot of things we could say about this phylogeny, but the first thing to notice is that Salmonella Dublin has a crazy strong geographic signal, probably more so than any pathogen I've actually ever worked with. So nearly all strains from North America cluster together in this yellow clade over here. And notably, nearly all strains from North America, especially the United States, they're multi-drug resistant and they possess this INC-AC2 plasmid. We can compare that to European Salmonella Dublin because nearly all European Salmonella Dublin belong to a completely separate lineage, and it's largely antimicrobial susceptible and doesn't have this INC-AC2 plasmid. And we found that nearly all of our South African genomes belong to their own distinct lineage that was totally different from the lineages circulating in North America and Europe. So I'll zoom in on that real quick. Basically what we found is that here in blue, and then denoted by this pink label here, basically there's a Salmonella lineage in South Africa, Salmonella Dublin specifically, that's been circulating among animals and foods in the country for decades. And occasionally this lineage does and has infected humans. However, there is some good news because it seems that Salmonella dublin in South Africa is largely antimicrobial susceptible. So it looks like antimicrobial resistance appears in this lineage only sporadically to our knowledge. So this is great news because unlike America, where every Salmonella dublin is basically multi-drug resistant, it looks like in South Africa, it's primarily susceptible to antibiotics. So that's good. However, one thing we did notice is that not all of our South African Salmonella dublin genomes cluster in this South African clade. Specifically, we had one rogue strain that was actually really closely related to Salmonella dublin from North America, specifically strains from the United States. So I informed my colleagues of this, and they were actually able to trace this strain to a poultry product that was being sold in a supermarket in Eastern Cape. So Atumeling actually was able to contact the supermarket, and the supermarket informed us that indeed the poultry had been imported from North America. They wouldn't say which country. I suspect it's the United States, but they did say it is from North America. But that's really cool because this corroborates exactly what our phylogeny told us. So if we had only looked at our small handful of South African Salmonella dublin genomes, we wouldn't have been able to conduct this investigation. So by incorporating thousands of publicly available genomes collected from different sources all over into the world into our analysis, we were actually able to generate a hypothesis regarding the origin of this particular strain, and this actually helped point the epidemiologists in the right direction for their investigations. So Atumeling thought this was really cool, so did I. And as it turns out, the South African government also thought it was pretty cool too. So Atumeling was actually able to get some more money to sequence more genomes and expand surveillance to foods that are being imported into South Africa, because it looks, according to our study, it looks like there's some strains that can come over to South Africa on imported foods. So this second case study involves poultry imported from Brazil. And Brazil, as it turns out, is a major global exporter of poultry, especially chicken. And South Africa is actually one of the top importers of Brazilian poultry. So those of you who are watching from South Africa right now, you guys actually import more poultry from Brazil than the entire European Union combined. I thought that was really interesting. But Atumeling and his team, they routinely test these poultry products for Salmonella. So they've been doing this for years. But recently they noticed that there's been an increase in the prevalence of a relatively rare serotype called Salmonella minnesota, and they found that to be quite strange. So Atumeling was able to collect 36 of these relatively rare Salmonella minnesota strains from frozen Brazilian poultry products, which had been imported into South Africa from Brazil. And he, again, was able to sequence their genomes. And Vishnu, who is a really awesome postdoc in my group, he was able to compare their 36 genomes to all publicly available Salmonella minnesota genomes. And there were 228 of these. So way fewer genomes to work with compared to Salmonella dublin, but still it is something. Again, like I said, Salmonella minnesota appears to be relatively rare, either that or it's just very under-sequenced, but definitely fewer genomes to work with, but we will make do. But here you can see a subset of the Salmonella minnesota phylogeny. Again, we have year plotted along the x-axis here. And then this heat map denotes the presence and absence of antimicrobial resistance genes and plasmids again. And then Atumeling strains, which were isolated from frozen poultry products that had been imported into South Africa from Brazil. So those are labeled with either supermarket, if the strain was isolated from a poultry product that was already for sale in a South African supermarket, or POE, which stands for port of entry. Basically, these strains were collected from poultry products that had just arrived to South Africa from Brazil, but they hadn't officially entered the country yet. So there's a ton that we could say again with this phylogeny. But the most interesting thing, in our opinion, was that we were able to identify a cluster of virtually identical Salmonella minnesota genomes isolated from Brazilian poultry that was already in South African supermarkets. And basically, all of these identical strains had a beta-lactamase gene called CTXM8. And this is super interesting because this is an AMR gene that is actually not very common in Salmonella at all. It's actually quite rare. So we were kind of like, this is really weird. How did all these strains get this super rare beta-lactamase gene? So after some more digging and basically analyzing millions of microbial genomes collected from all over the world, we were able to come up with a hypothesis. So our hypothesis is that this CTXM8 gene, while it's really rare in Salmonella, it's actually a bit more common in E. coli. Like, it's not super common when you look at all E. coli genomes. It's still quite rare, but it's definitely more common in E. coli than in Salmonella. Not only that, this specific gene seems to pop up in Brazil quite a bit more than a lot of other places. So based on our analysis, our hypothesis is that in Brazil, this Salmonella Minnesota lineage was able to acquire the CTXM8 gene, so this rare AMR gene, from E. coli strains that had it. And then the CTXM8 harboring Salmonella Minnesota strains basically hitchhiked to South Africa on some frozen poultry products, and that's where Itumeleng and his group picked them up. We can't know for sure if this is what happened. Of course, it's just a hypothesis, but overall, I think it's a really cool story of how genomic data can help us detect events like these. And also, I think it's an important lesson for how events on the other side of the world can end up affecting all of us. But now that I've made you guys like sufficiently depressed about antibiotic-resistant bacteria hitchhiking into South Africa, let's switch to the more optimistic topic of novel antimicrobial discovery. So as we know, antibiotics are really important for treating bacterial infections. And many critically important antibiotics, including all of the examples that I list here on this slide, they are derived from what we call microbial secondary metabolites. So what are microbial secondary metabolites? Well, secondary metabolites are compounds that microbes produce that aren't directly involved in their growth, development, or reproduction, but these compounds play really important roles because they allow the microbes that produce them to interact with each other, as well as with their environment more generally. And as we already know, many critically important drugs, including many antibiotics, are or have been derived from microbial secondary metabolites. And again, these are just a couple of my favorite examples. However, not all microbial secondary metabolites are good guys. Some secondary metabolites are toxins or carcinogens. And these are just a couple, again, of my favorite examples, but there's thousands more out there. But whether they're really good or really bad for human health, we have a pretty big incentive to try to find novel secondary metabolites. And as it turns out, the enzymatic machinery responsible for the production of many microbial secondary metabolites, it's encoded by physically clustered groups of genes within microbial genomes called biosynthetic gene clusters, or BGCs. So if you can kind of see where I'm going with this, there's a huge incentive to mine microbial genomes and metagenomes for novel BGCs, because these novel BGCs might be responsible for the production of novel secondary metabolites. And these novel secondary metabolites might have really cool roles in terms of like how the microbes respond and adapt to stressors in their environment. But more importantly, they might have really cool industrial or medical uses, such as being novel antibiotics. So to that end, during my postdoc, my collaborators at EMBL and I developed a tool to basically find novel biosynthetic gene clusters, or BGCs, in microbial genomes and metagenomes called Gene Cluster Prediction with Conditional Random Fields, also known as GECKO. I don't have time to go into the details, but GECKO works by taking a prokaryotic genome or metagenome assembled genome as input. It then identifies genes and protein domains in that genome. And then GECKO identifies regions of that genome that have a high probability of being a BGC. And it does this using a type of machine learning model called a conditional random field, or CRF. And then as a final step, GECKO uses a random forest classifier to predict the biosynthetic class of a BGC. So when compared to other machine learning-based methods on a BGC discovery task, we found that GECKO was by far the most accurate. So for example, this is a precision recall curve here for GECKO in green, Deep BGC, which is a competing machine learning method in blue, and then another machine learning method in orange called Cluster Finder. And if you've never seen a precision recall curve before, don't stress, basically the greater the area under the curve, the better the model. And as you can see, gecko outperforms the other models. Not only is it more accurate, but gecko is also faster than previous methods. So for example, here you can see how long it took for each of these tools to identify BGCs in over 10,000 diverse bacterial genomes. You can see each tool along the y-axis here, and then the total time it took in hours for detecting BGCs in this set of 10,000 plus genomes using a single CPU. And it's pretty clear to see gecko is like 4 times faster than deep BGC and 10 times faster than anti-smash. And this is actually a really exciting benchmark for us because it shows that we actually developed a method that can scale to massive data sets that previous methods struggled to scale to. And I'll come back to that. But also because conditional random fields, the type of machine learning model that we used, because they're interpretable models, we can actually look under the hood of gecko and see what it actually learned and deemed important for biosynthetic gene cluster discovery. So for example, this is just a simple box plot of the weights that gecko assigned to all of the protein domains in its model. And we can see here that some protein domains received really high weights, meaning that they're strongly BGC associated. They're very important for detecting BGCs in microbial genomes. And some of them were pretty cool and made a lot of sense to us. For example, sublancin received really high weights. And sublancin basically is known to display antimicrobial activity against some gram-positive bacteria. So that was actually really cool because gecko learned that this antimicrobial associated protein domain is important for BGC detection. So that was really promising. However, many of the most important BGC-associated protein domains were what we call domains of unknown function, meaning that we have literally no idea what they're doing. And we actually found like a couple dozen or so highly weighted domains of unknown function, which are apparently really important for biosynthetic gene cluster prediction. So hopefully in the future, maybe people might be able to figure out what these protein domains are doing and why they might be important for secondary metabolite production. But since we developed gecko a couple of years ago, we've actually been using it to mine massive genomic and metagenomic data sets trying to find novel BGCs, particularly those that might encode novel antibiotics. So for example, we collaborated with some ocean microbiome researchers at ETH Zurich, and we basically mined their set of 34,000 ocean metagenomes trying to find novel secondary metabolites. We also applied gecko to all microbial isolate genomes in the Progenomes 3 database. So that's like almost 1 million genomes just there. And then most recently, we actually used gecko to mine the entirety of all the bacteria, which is a data set of nearly 2.5 million bacterial genomes. So basically, this is pretty much together all the prokaryotic isolate genomes ever sequenced and available publicly at the time. And we've made all of these predicted BGCs available to the public with the hope that anyone in the entire world who's interested in antibiotic discovery can use them. And currently we're trying to build an interactive atlas to make these BGCs easier to search and play around with and visualize. So that will be available to the public as well in the future. But right now they're kind of just deposited in the databases and not as pretty. But if you're interested in antibiotic discovery, check out all the bacteria and Progenomes 3 because you're more than welcome to download all of our predicted BGCs. They're there for anyone in the world to use. But now we're kind of running into a new problem now that we have all of this predicted BGC data. How do we actually make sense of it? Basically, how do we help biologists and chemists find their needle in a haystack BGC, so one that might be a novel antibiotic? So to explain this problem, let's pretend that we have millions of predicted BGCs, represented by this collection of gray arrows. And let's pretend that this pink BGC here is a totally novel BGC that equips some bacteria with the ability to produce a novel antibiotic. So we basically want to find this pink guy here out of millions and millions and millions of BGCs at our disposal, some of which might be novel and unlike anything we've ever seen before, others that might be responsible for the production of a known secondary metabolite. So how do we do this? How do we find novelty in an endless sea of candidate BGCs? So one way of tackling this problem is to compare our unknown BGCs to a set of known BGCs. For example, some experimentally validated BGCs that we know produce a specific secondary metabolite. we can then use some sort of clustering algorithm to cluster our unknown BGCs along with these known BGCs into what we call gene cluster families, or GCFs. So by doing this, we can assign predicted function to unknown BGCs. The rationale here being that if you belong to the same gene cluster family, or GCF, you're likely to produce a similar metabolite. So during my postdoc, Martin Lerald, who is a really awesome PhD student, he and I developed our own BGC clustering method called Iterative Gene Cluster Analysis, or IGWA. This is the overall workflow. I'm not going to go into it because it's a little technical, but I figured I'd end with the cool things that we've actually been doing with IGWA, specifically what my PhD student Josephine has been doing. Specifically, Josephine used IGWA to cluster over 2.8 million biosynthetic gene clusters from 1 million prokaryotic genomes. So all the BGCs that we detected in the Progenomes 3 database using Gecko. And here is a UMAP, so basically like a two-dimensional visualization of all of these BGCs clustered into gene cluster families. And we colored them by their predicted biosynthetic class. And I don't really show it here, but the vast majority of these gene cluster families, the vast majority of them don't contain any experimentally validated BGCs, meaning that there's so many BGCs out there that experimentalists have just never even touched. So currently my team is looking into some of these that we hope have antimicrobial properties, but we only have very preliminary data so far, so I won't show any of it because we're not sure yet, but that's something we're working on and it's ongoing work. But it's really exciting. We found a ton of new novel potential secondary metabolite encoding biosynthetic gene clusters. So because I'm like almost out of time and things are winding down, I figured let's go to the conclusions. So in conclusion, we can leverage publicly available genomic data and metadata to help us in the fight against antimicrobial resistance, both from a surveillance and from an antibiotic discovery perspective. But it's also important to note that genomic data alone only gets you so far. To actually make sense of all of this data and use it to its full potential, we all need to work together. So microbiologists, clinicians, epidemiologists, bioinformaticians like myself, people in the social sciences. Basically, we all need to work together, and collaboration is essential if we want to tackle this massive problem. So before I wrap up, I'd like to give a special thanks to all of my collaborators, especially Atumelang and Tendo and Atumelang's team. All the pathogen surveillance work would not have been possible without them. They led and designed those studies. They're the stars of the show. I just am lucky enough to present it today and played a small role in the analysis. Also Vishnu, who is a postdoc who worked on a bunch of the analysis of Salmonella Minnesota data, and then Josephine and Martin and Georg, who were involved in all the biosynthetic gene cluster projects that I work on. And of course, thanks to my whole group and thanks to my funders for actually paying me so I can be here today with you guys. And thank you all so much for listening. It is such a pleasure to be here. And hopefully I lived up to the wonderful introduction by Dr. Lakota. And thanks for listening.
Thank you so much for that wonderful talk. I think we'll now move over to the Q&A session. Feel free again, guys, if there's anyone who has some questions, they can still post them on the chat. I have a few of them that I can just post out from the platform. I think the first question that we have here is, does Illumina data capture the entire plasmid profiling of the Salmonella?
Oh, sorry, can you say that again? I missed it.
There's a question here on the chat that say, does Illumina sequence data manage to capture all the entire plasmid profiling of the Salmonella?
Wonderful question. No. So you can get, with Salmonella, we're lucky that you can look for replicons and it seems to be pretty informative. But 100%, if you want the, I guess, most accurate representation of what plasmids are in there, you definitely need long read sequencing. And that is like the way to go. Sadly, these were just short reads that we had. And yeah, we wish we had more because we had some cool hypotheses about Salmonella Minnesota possibly having an integrated plasmid, but we'll never know for sure. You need long reads. So good question. Short reads, you can get so far, but there's a level that you can't get over without long reads.
Okay, thanks for that feedback. Another question from the floor, I think. We have, how do you know that Salmonella Minnesota plasmid, particularly the BLAST CTX-XM8, resemble those of E. coli from Brazil? Any plasmid experiments that have been conducted on these genes to make that hypothesis?
So exactly. So this was basically what we did is we found that this BLAST CTX-M8, we couldn't get a perfect plasmid because we just had Illumina data, but we found that at least the entire contig it was on, the surrounding contig, is like an identical match to a bunch of like similar plasmids, including closed plasmids from E. coli. And basically, we can't know for sure that it came from Brazil, but it seems likely that that's where the strain originated, given that we had isolates. We know the isolates came from Brazil, and it basically based on metadata, it seemed to pop up there more often than not. Yeah. But again, hypothesis for sure.
Okay. Development. Okay, I see. Okay, I think another question from the platform is, any E. coli strains that could have also been screened from South Africa chicken or poultry, perhaps to link this correlation, as the same chicken samples might have carried the BLA CTX-M8 gene?
That is such a good idea. Sadly, I did not have access to any E. coli genomes. 100%. I feel like a Tumalang would have that, and there is a chance he may have sequenced them. That's a really good idea, though. You could see if it's also in E. coli as well in South Africa. Yeah, no, so we didn't look at that, sadly, but there's a chance a Tumalang may have or may be looking into that.
I'm not sure if it's my network. Okay, thanks for that. Um, Another question on the platform. It says, has Gecko tool been applied in other sources just rather than just soil, rather than just the method, the water, or just the waste wood? So let me just repeat the question. It says, has Gecko tool has been applied in other sources besides the ocean water?
Great question. I didn't show it today. Actually, the first thing we applied it to was basically every gut microbiome metagenome and isolate sequence ever sequenced, because my postdoc advisor is a gut microbiome person. So that was why. So we actually were able to find a new secondary metabolite produced by Bacteroides there. We still don't know what it's doing, but that was actually kind of its maiden voyage. But I think for drug discovery, I think soil microbes and possibly like ocean microbes, the more environmental sources, you have a better chance of finding like an antimicrobial. So, but yeah, we've applied it to pretty much every genome and metagenome that exists now. The isolate BGCs are all out there. The metagenome BGCs we should be releasing this year, for every basically metagenome and every environment that is in the MO2's database.
Okay. I don't see any other questions from the floor. Perhaps maybe if they've got any other questions, maybe they can follow up by just emailing you. I think I will just end here for the session because there isn't any more questions that are actually posted on the chat. Okay.
Thank you guys so much, and
What do you-
thanks for the great questions.
Thank you so much. I think I'll move over to Mr. Mahlatsi.
Oh, thank you. It's a wonderful, wonderful talk. Can we move to Prof. Carlos doing the closing?
Good morning, everyone. Colleagues, participants, distinguished guests. This is a very special occasion for us. At the Northwest University, the subject group microbiology took the initiative in starting the first World Antibiotic Awareness Week conscientization in 2018. It's now 8 years later and we're still going strong. So let me start by thanking Mr. Abram Matlatsi for directing the program today. He was part of the very first team. Thank you, Abram, for your continuous support. Professor Lesego Molaletom, she was currently director of the School for Biological Sciences, where microbiology is situated. She was also there in 2018. So thank you for the opening and for keeping the reins and ensuring that these events continue to take place. Professor Lakota, thank you for liaising with the speaker and for the great introduction that you provided, as well as for facilitating the question and answers session. Then a special thank to Dr. Laura Carroll from Umeå University in northern Sweden. And those of you that didn't know, that is the home of Nobel Prize winners. Thank you for a mind-provoking talk about how modern technology can be physically used in today's age to understand the presence or the present scenario around AMR, and as well as predicting futures and potential future solutions that we can already start implementing in terms of our stewardship. Mrs. Lee Shanaka and the team, thank you very much for the organization and everything that went into ensuring that this is a successful event. Then the social media, Janine Viljoen, as well as Mr. Paul van Zyl for Mr. Paul van Zyl Company did the Zoom recording as well as the YouTube channel. So thank you all for your contribution and ensuring that the event was successful this morning. So thank you. That brings me to the end of the closing remarks.
Thank you. Thank you very much, Prof. Can we take a few seconds for the event survey, please? We'll take you a few seconds. Thank you very much, everyone. Thank you for attending. We'll see you on the next one. Thank you.